उसमें मैंने कुछ गलत पेस्ट कर दिया. It can be a copy paste error. It can be an error simply due to data fabrication. You don't know. You have to go back to the root cause. Sagar PCR parameter design. Before you okay, I will discuss few things. How you can do in silico PCR, how you can do check your PCR primers. And I will just focus on one tool, that is Primer Blast. It's a very wholesome tool. आपको बहुत कुछ मिल जाएगा, background correction, this, that. And first we'll go through the basics. PCR design PCR. One gene. But if you see in early 1980s, people had a very good understanding how DNA replication works, right? And how does DNA replication work? You have double-stranded DNA, which has to be melted or unwound. There you have an enzyme, primarily a polymerase. That enzyme has multiple parts. One of it produces a small fragment of DNA, right? Small fragment of RNA बनता है, छोटा सा एक primer बनता है, primase enzyme से. So you need that, and then your DNA polymerase starts synthesizing. Then you have this lagging strand and leading strand and Okazaki fragments. So this was sorted. Now, nobody could actually think or wonder, and how did people amplify genes? By that time, we did start amplifying genes, and we were amplifying genes using a very basic technique that even today we use. That is, how would you amplify a gene if you don't have a PCR? Not for expression studies, but if you have to make copies of a gene. In the year 1983. Somebody called vectors. Cloning. Cloning was our initial photocopy machine. So, किसी को भी अगर किसी DNA को amplify करना होता था, they used cloning as a technique. But cloning is a long process, right? Or even we needed to clone one gene. But if you see, we had made lots of good discoveries by then. We had already synthesized artificial insulin. So we were making progress. One big challenge was how to do DNA replication in vitro. And that is where Carey Mullis comes in. And Carey Mullis was not your ordinary scientist. He was a maverick. You know what the English term maverick means? A guy who is extremely talented, or somebody like Iron Man. Right? Extremely talented, but he won't listen to anybody's rules. He will make his own rules, and that's why he was not a very loved figure among his colleagues. Anyway, the maverick he was, when he was driving down from his lab to his hometown, it was a weekend perhaps. He was driving in his car. And as it often happens with scientists, with every human being, if you're thinking about something, आप किसी problem के बारे में सोच रहे हो, what happens? आप कुछ भी काम करो, दिमाग के पीछे वो चलता रहता. And suddenly, he had this idea that's floated. होता है कभी-कभी हम लोग कुछ चीज पे बहुत सोचते हैं. And suddenly, everything becomes clear. So that happened in the year 1983. But PCR as a technique was not very robust in 1983. He gave us the fundamentals. There is a very good paper in National Geographic channel. एक काफी अच्छा article है that tells more about PCR, but since today's focus is more on primer designing, but all of you can look up how Carey Mullis. It's a very interesting history. काफी सारा personal ego clash, funding agency के साथ लड़ाई, अपने दोस्तों के साथ लड़ाई. He was thrown out of his own lab. Then he comes back to his own lab. तो काफी high drama, high octane drama. You can make a good biopic out of it. ये सब होने के बाद, by early was an established technique. And what do we need for this technique was another invention was the invention of Taq polymer. The initial parts when Carey Mullis thought of PCR, the biggest challenge was the thermolabile nature of DNA polymerases. Since it's an enzyme, it was very difficult for us to use that enzyme at that specific temperature. So initial days of PCR, हम लोग क्या करते थे? We heat and denature the DNA, cool it down, add enzyme, then again denature, then again add enzyme. So at every step you have to add enzyme because the moment you heat it at 94, आपका enzyme खत्म. फिर आप उसको ठंडा किया, annealing में लिया, थोड़ा सा enzyme लिया. But in principle, they were waiting for one discovery, an enzyme that could tolerate the heat. That came in the form of Thermus aquaticus DNA polymerase. Again, the reason why we should study metagenomes. So it was again a thermophilic bacteria that they could find in the hot water springs, and somebody had this, again, an idea that if a bacteria can survive in this heat, most probably the enzymes that this bacteria produces will also be thermostable. So it's an important component. And then it was already sorted by Carrimulis that you need to add DNA nucleotides or DNTPs. You have to have a template DNA. You have to have short-stranded DNA, 16 to 50 nucleotides. Again, this was a synthesis. We need to thank the chemists. अब जितना बायोलॉजी के अंदर घुसते जाओगे ना आपको लगेगा कि all the prime discoveries were made by chemists and physicists. We are more on the application based. So chemists invented how to synthesize nucleotides artificially. Artificial gene synthesis was discovered again early 1970s. So all these things in early 1991-92, Carrimulis had developed PCR as a robust technique. People immediately understood the importance of PCR, and Carrimulis got a Nobel Prize in 1993. देखो आप Nobel Prize में अगर trends देखोगे, you would see technologies which got a Nobel Prize very fast had shown lots of promise. There are two ways of getting a Nobel. Either आपने कुछ discover किया and you went ahead of your time, you discovered it. लोगों को 10-20 साल तक पता नहीं चला इसका importance क्या है. After 40 years, they realize, hey, this is the importance. And then again 10 years later, they give you a Nobel. By that time, you're 90 years old. And if you die, the Nobel goes. Else, like Carrimulis, you discover something which is extremely important, that is a breakthrough that changes the way we do science, and you get a Nobel Prize which is very fast. So, we were talking and primer annealing. So, this is a basic step and of a polymerase chain reaction. You usually have 30 to 40 cycles. The first part that is specificity. All primers should be specific to your DNA template. This is the first and foremost thing that you have to remember. आप primer designing के बाकी rules भूल जाओ, इसको मत भूलना, right? Because this is where the game begins. बाकी सारे जो है, these are all tinkerings, right? You make a rough draft of anything, and then you start polishing. So this is that rough cut diamond specificity. Your primer should be specific to the template. आप जिस gene के against आपने primer बनाया, उस gene के साथ आपका primer का एक, actually दो forward and reverse के लिए दो binding points होने चाहिए. That will define the boundary of the area which you will be amplifying. Right? So before you start designing your primers, don't reinvent the wheel. What is reinventing the wheel? This is a idiom in English. Don't reinvent the wheel. Don't. They're very good, and these are active databases. जहाँ पे आपको जाके specify करना होगा कि आप किस gene के against ढूंढ रहे हैं. And if you're working with a modern system, like you're working on mouse, right? Musculus, Ratus novagic.